very much, Emma, and it's a pleasure to be here. See so many people we've been liaising with and helping over the years. It's a real privilege to meet you all. This morning we heard some marvellous talks, and I don't know quite how I'm going to follow that. The remit I had was to discuss really the genetic variants and what you might have been told in your genetic report if you've had that done, and then go on to explain what might trigger an episode of aHUS. So we'll discuss what genes cause aHUS. We'll describe the different types of variants you might have been told about. We'll go over the genetic report. We'll discuss why some changes may be significant and why they might not. We'll discuss what we're doing if people don't have a genetic variant, and then we're going to discuss triggers. So, you heard from Professor Goodship about adverse modifying variants earlier on and how we're increasingly starting to understand them, but it's not the whole picture. To get disease, we believe you need both one of these modifying variants, a trigger, and a predisposing significant variant. And that's what I'm going to focus about in the first part of the talk. So, as you heard from Kevin, complement is an immunological mechanism to kill bugs. When you get a bug, be it bacteria, virus, parasite that invades your skin, it triggers the complement system. And the complement system, as Kevin said, is an amplification loop. It really accelerates. It has evolved over millennia to try to kill bacteria. And to do that, it needs to work very quickly. And because of that, you have this positive amplification loop. Now, what that does is it produces things that kill bacteria. Inflammation, it covers the bacteria in something that says, kill me, and it produces a pore that lyses bugs. So complement is very good at killing bugs. And bugs are very good at activating the complement system, and that will become important when we discuss triggers later on. Now, as we've said, as Kevin kind of said, there are parts of the complement system that act as an accelerator. So if you look at this bit here, this is the accelerator of the complement system, and these are the brake that we talked about. So when you get a bug, it triggers this complement system. And what you want is for the complement system to be activated and to kill those bugs. But what about the collateral damage? When you've got all this complement activation, how do you protect your own surfaces from collateral damage? And you do that by putting the brake on. And these are complement factor H, factor I, and membrane cofactor protein. These are very much the brake of the complement system, counteracting the acceleration that these bugs started. What these do is these come in and they cleave one of these complement proteins, C3b, to the inactive form, and really stop the complement system stone dead. So it's protecting your kidney once the bugs have been killed. And for complement-mediated aHUS, those are the 5 proteins you really need to think about. So there are 3 proteins that act as brakes that have been shown to be involved in aHUS. That's complement factor H, complement factor I, and membrane cofactor protein. And there are other ones, complement C3 and factor B, where you have the accelerator stuck on. So those are the main 5 proteins in complement-mediated aHUS. So, do you have a mutation? Well, many people in this room will be told they have a mutation. Some people have been told we can't find a mutation, and other people may have been told that they have a genetic variant of uncertain significance. I think Tim kind of briefly covered this, and most people will know the term mutation, and I think the terminology is very much changing now. Historically, you might have heard the term polymorphism, and this was thought to be common, functionally inconsequential, and didn't really have any role in disease. Whereas a mutation was thought to be rare, functionally important, and disease-causing. Now, as we know more about genetics, we find polymorphisms that can cause various diseases and mutations that have no effect. So now we try very hard not to use the term mutation, and we use the term rare genetic variant. And then we specify if that genetic variant is significant. So, this is James Watson. Now, he was one of the first people to have his whole genome sequenced. And you'll know him from being the first person to define the nature of DNA. And when he had his genome sequenced, so all the genes in the human body, he had genetic variants in a gene called ERCC6. Doesn't matter what it is. And if that was significant, he should have been a dwarf with a small head and died early. He had another mutation in a gene called myosin 7A, which causes, if it was important, would cause him to be deaf and blind. And James Watson is none of those, even if he's out of favour at the moment. So, not all spelling mistakes in genes cause disease, certainly in aHUS. Now, there are some other genetic conditions where if you have the genetic disease, you have the genetic change, you will get disease. In cystic fibrosis, it's a lung disease. Many of you have heard about Gordon Brown's son being diagnosed with this. If you have this change, in America, 90% of the people with cystic fibrosis, it's caused by this, you will get disease. This means that genetic testing is much simpler in some genetic conditions than atypical aHUS. Now, I'm only going to cover factor H, but the same can be said of all the complement genes. These are all the genetic variants we have found in factor H in atypical aHUS. Now, not all of these are functionally significant. So when we read people's DNA and we read the instruction manual, how do we know if these changes we find are important? Well, stretching out Tim's analogy of Lego, and I would like to stress there are other plastic construction kits available. I'd like to introduce you to our Factor H F1 team with our driver Jensen aHUS. Appropriate logos you note. And there are an instruction code for this car. And this car is, we're called Factor H. And here's the instruction here. Now, you've got an engine which powers the protein, and you've got a steering wheel which tells it where to go. Now, this was kind of what happened when I was building Lego when I was young. Oh, Dad, you've put the coffee cup right over the instructions. And now there's a problem. Now, Jensen's a smart driver, and he's looked and thought, oh, there's no engine. It's not going to go anywhere. It's not going to do anything. And something else is missing. And the steering wheel's not there either. So these instructions, your genetic code different things. And if the spelling mistakes is a really important bit, the protein won't function. Now, this is what my Lego tended to look like. Not quite as colourful and not quite the right shape. But you can see it's got an engine and it's got a steering wheel. It might look a bit different and the proteins might be a bit different, but it'll still drive. And that's what we need to find. Those other bits, provided they can join the engine and the steering wheel together, your protein will work. Might not look as pretty, but the protein will work. So when we find a genetic variant, a spelling mistake in the instructions, we need to work out, well, is it going to make the protein non-functional? So, factor H. Well, there are mutations in factor H, and there are two key areas of factor H. Number 1 to 4, that's the engine of factor H. That is the bit that cleaves C3b and stops that positive amplification loop. So essentially, key to working. If 1 to 4 doesn't work, the rest of the protein won't work. And 1920 is the steering wheel. The steering wheel tells it where to go. So, if you look at where the mutations or genetic variants are in factor H, you can see that the vast majority are actually in the steering wheel in this 1920 region, and that's the area of factor H that targets the kidney. So in aHUS, most of it is due to the fact that the protein can't get to the right point. There are a few other mutations in here that stop C3b being cleaved. There are other mutations, and they're in regions that link the engine and the steering wheel, and we don't really know what they do. So what do we do in that sort of situation? If we find a variant in a bit of the protein, we don't know what it does, how do we know if it will be functionally significant? And this is something Tim and I ponder over every Tuesday when we get these results back. So when we get this variant, we can say, well, how common is this variant? If it's common, it's not likely to cause disease. Does it run in a family? Tim told you at length how families are critical in understanding the genetics of aHUS. If a genetic variant runs in a family, it's highly likely to cause disease. Is the protein produced? If you get the instructions out and there's one of those stop instructions, the protein won't be made. So if you measure the level, it won't be there. And the other thing we can do now is look at protein prediction of what this will do. That's not very reliable. So, one of the advantages we have in Newcastle is we have a research laboratory stuck onto the side. And what we can do is we can look at the structure, we can see how the proteins interact, and we can actually set up bacteria to make factor H and factor I and the other ones, and do experiments to see if these are important. So first thing we can do is we can look at the structure of the protein. Now this is an NMR machine. It's you might have an MRI yourself. Essentially it's a big magnet that looks at very small proteins. And what we do here is we put our very tiny protein into this magnet and we say, well, does it look normal? And what happens when you put this in there is you get something called an HSQC. I can't say what it stands for either, but what it actually is, it's a fingerprint of the protein. So every protein has a fingerprint and it changes. In these cases it doesn't change very much, but if it was to change a lot, we would know it was functionally significant. The other thing we can do is we can look at how proteins interact. So this is the engine of factor H. And all those red dots are mutations or genetic variants. This is C3b, and you can see how it interacts. So if one of these red dots comes up against C3b, you can be relatively sure that it's going to play some role in function. Now the other thing we need is an Edwin, and here's Edwin. Edwin's going to speak to you next, and what Edwin does is he makes proteins in this. So this is a fermenter, and he's not fermenting alcohol. What he's actually fermenting is factor H. And what Edwin does is he makes recombinant factor H. He uses yeast to make a normal protein and a mutant protein and sees if they function. And this is one of Edwin's experiments, and you can see that this is the complement system functioning nicely. It doesn't actually matter what it is. But then when you add normal factor H, this is the brake, you can see it comes screeching to a halt and immediately stops. If you add a mutant factor H, then nothing happens. So what Edwin did here was to show that this genetic variant in factor H didn't work. The brakes failed. So the other thing you need is a Holly. Now, Holly makes different bits of factor H, and what she did here was to look at the steering wheel of factor H. And what you can see here is that when you looked at the steering wheel, these rare variants in factor H didn't work. That would give you the impression that every variant we looked at doesn't work. But in actual fact, here is an experiment by Santiago's Spanish group. And what he shows here is that these variants make no difference to function. So you've got many variants you know will function properly, but you've got these other variants that won't. So it's very important when we look at your genetic information to be able to interpret these. And sometimes we just don't. Because unfortunately, to do these experiments that Edwin and Holly have done takes 2 to 3 years for just a few variants. And you've seen there are hundreds. It's costly, and there are very variants. So what if I don't have a genetic variant? Now this is a Rachel, and what Rachel does is she looks at people without any known mutations, and she looks at the whole genome. So she's doing what they did years ago with the Whole Genome Project. So just to give you an idea of that, there are 6 billion base pairs. That's 857,000 pages of text, and that's 28,000 Shakespeare plays. And if you like Shakespeare as much as I do, you know Rachel has a pretty arduous job. In that 28,000 Shakespeare plays, there's 30 to 40,000 genes, just for one person. Now, to show you what that looks like, this is a factor H gene. It's not actually the factor H gene, it's page 1 of the factor H gene. There are another 100 pages like this, and then this. And I don't know if anybody spotted the mutation in there. There we go, in there. So that's what Rachel's looking for in every single gene in the body, to see if she can find any spelling mistake that might cause aHUS. Now, this has only really been possible in the last few years. When the first Human Genome Project was done, it cost over £100 million to sequence somebody's genome, and involved centres all around the world. Today, you can do this for a couple of $1,000. So this is what Tim meant by state-of-the-art sequencing. You've seen this pretty much in every talk. So this is what Rachel did. So she went through those 20,000 pages of Shakespeare or whatever for every single individual in the families in Newcastle. And what she found was that the vast majority were complement mediated. But we still don't know about 30%. And she had identified a few new ones. So hopefully the next time we have this conference, we'll be able to tell you about it. And Paddy, who's in the audience, will hopefully be taking on this project next. So it's not quite as straightforward as just having a predisposing significant variant. When we get this change, we need to show that this is important for you. So you need a rare genetic variant that is functionally significant. You need one of these modifying variants, but you also need a trigger. Now, what are these triggers? So this is a very busy slide. And this is a list of triggers of aHUS. And what you notice is, from here to here, it's all bacteria, viruses, and parasites. That's not particularly surprising. I said at the beginning that bacteria, parasites, and viruses are very good at stimulating the complement system. It's putting the accelerator on. And that's good. That saves you from getting an infection. But the problem is, if you don't have an adequate brake, once the complement system has dealt with the bug, you can't stop the collateral damage. Another one is pregnancy. I'm not going to talk too much about this, but Edwin's going to talk in detail about our experience of pregnancy-associated aHUS. And really, we're not entirely clear about why pregnancy triggers aHUS. In the postpartum, you can get infections, you can get postpartum bleeding, two things we know activate complement, and there are also physiological and hormonal changes that probably have a role. Many drugs will also cause aHUS. Now, some of these may well trigger the complement system, but not necessarily all. So the mechanism of some, quinine for example, is known to trigger complement through antibodies. In others, say mitomycin, it's direct endothelial damage. There are other autoimmune triggers. Most of these, again, trigger the complement system. So the triggers of aHUS are, as you would expect, they're mostly infectious and they mostly start the complement system up. And that is our current understanding of the triggers of aHUS. So what have I said? There are multiple genes associated with atypical aHUS. We've discussed them. There are non-complement mediated disease genes, most commonly DGK epsilon. And the genetic variants we see in individual people and families tend to be personal to you. And sometimes we just don't know if these genetic spelling mistakes we find are significant. Often we do, but not always. And that these spelling mistakes, only the functionally significant ones will predispose to disease. You get these modifying common variants that Tim discussed, and only when you've got the functionally significant rare genetic variant, these SNPs, and then you get a trigger will you develop aHUS. And I'd just like to thank everybody in my group for their work over the years. And I'm happy to take questions.
Okay, thank you very much, David. I'd just like to take this opportunity to thank David for all the work he's done in setting up this conference and coordinating all of the speakers who won't give their PowerPoints in advance, apparently, ever. Any questions? Wow, it was so comprehensive that they don't have to answer any question. Oh, here we go.
If you know which mutation you've got, can you get any insight then from knowing whereabouts it is or anything like that?
Yes, so what Tim and I normally do on a Tuesday is we get these genetic reports back, and if it's in the engine and it's a big change, and we do all these computational tools that we went through, it's highly likely to be significant. We've made a lot of these mutations, so sometimes we're able to look and say, it's very close to a mutation that is significant. We're able to say, this is highly likely to be significant because it is in the engine or the steering wheel. If it's in the bit that links the engine to the steering wheel, we're not entirely clear. So sometimes we don't know. Yes?
You've had some gene work on your bloods quite a few years ago.
Yeah.
2005, 2004. Would it be worth looking at that again because of the technology moving on and understanding?
Yes, we very much try to do that, especially if you're going to get transplantation. Anybody that's being listed for transplantation, we'd want to go back and screen all the genes we know about. Where it's going to cause a clinical decision process being triggered, we'll go back and screen those genes. If that's not the case, you can certainly ask for that to be done, and the results fed back. If you ask your clinician, they can speak to us and say, what have you done? Should we be doing any more? And this will be a process that evolves. We've talked about the novel genes we've screened. Many people will not, most people would have had them screened, and once we release these and we're sure from other European cohorts that they are indeed true, we'll go back and introduce this to the screening service. It's very important that you go back and always update, because clearly if you presented 30 years ago with Professor Goodship, we didn't have these technologies, so you might only have had a bit of factor H screened. But we need to go back and look as we learn more and more. Yes?
The third circle in the diagram.
Yes.
You talked about the first one, which is a mutation.
Yes.
Second is the trigger. The third circle, didn't quite understand what the significance of that.
Okay, so these may have been termed a polymorphism in the past. So these are something that are very common. So what Tim showed was that for one of them, if you have a haplotype of MCP, if you get a bit of DNA, if you've got aHUS, 40% of the people will have that. And it's very common because 20% of the people in the normal population will have that. So it's a common thing that alters your level of complement regulation. On its own, it's not sufficient to cause disease. It's just, it's a little tweak on the brakes and the accelerators of the complement system. It's not like a mutation that has an all or none effect. So most of the mutations or rare genetic variants with functional significance will wipe out function completely. In these haplotypes that Tim talked about, the effect is small, but it's additive as well as a mutation. So it's just a little tweak on the brakes or the accelerator that just push you over the edge a bit, but only once you get this trigger. So it's multiple genetic factors plus this trigger that activates the complement system. Yes?
If you have an unknown mutation,
Yeah.
is there a chance that the inclusive wouldn't work?
Yes. So from what we know was, in the initial report of people with DJK epsilon mutations, one person was on eculizumab and developed a recurrence. We do not know the exact mechanism of action of DJK epsilon yet. There's a lot of speculation, but certainly in our experience, if you've got a DJK epsilon mutation, you do not respond to eculizumab. Clearly it's early days, we need much more information, but yes, there is the possibility that if it's not complement mediated, you will not respond to eculizumab. Thanks very much.
Okay. Thank you very much, David.